there was a paper a couple of years ago in which they proposed changing the name from canine distemper to carnivore distemper, but as we've subsequently found, that would still not cover all the different species. Canine distemper is actually a remarkable virus in that it can infect everything that is exposed to the virus. It's an extremely broad tropism virus.
Hello, and welcome to Contagious Thinking, the podcast from the MRC University of Glasgow Centre for Virus Research, brought to you by its very own staff and students. I'm Joanna Crispell, a PhD student at the CVR and your host for this episode. Here, fellow PhD student Yasmin Power and I talk with Dr. Brian Willett, who is Professor of Viral Immunology here at the CVR. In today's episode, we will be discussing Brian's recent publications, which involve 3 different viruses of the morbillivirus genus. So I thought I would give you a quick overview of these viruses before we begin. Canine distemper virus, known as CDV for short, is an inappropriately named virus, as it has been found not only to infect dogs, but also a very wide range of animals such as pandas, coyotes, ferrets, and large cats, to name a few. It causes gastrointestinal and respiratory disease and is a threat to conservation of already endangered animals such as tigers. Another virus, pest deputy ruminant virus, known as PPRV, is highly contagious and infects small ruminants such as sheep and goats. Cattle and pigs can also be infected, but don't develop any clinical signs. The disease is widespread in many parts of the world, including large parts of Africa, the Middle East and India, and is spreading into other countries. The infection of animals in these countries is especially serious as they are so important to people's livelihoods, and an outbreak of disease can lead to high economic burden. A very famous morbillivirus is known as rinderpest, a virus that was declared as successfully eradicated in 2011 after a global effort. It became the second virus ever after smallpox to be declared eradicated. Rinderpest virus infects cattle, buffalo and yaks, but animals such as giraffe, wildebeest, kudu and various antelopes can also carry the virus. The more virulent strains of the virus could lead to mortality in 80 to 90% of animals infected. So, that's a short introduction to the 3 morbilliviruses we will discuss in this episode. Now, let's find out more from Brian about the assay developed in his lab for the detection of neutralising antibodies to these viruses and the interesting results that they have found.
So just to start off with, can you give us a little bit of background about what you and what your group study?
Well, I actually joined the vet school in 1989 to study feline leukemia virus with Oz Jarrett. Then I moved on to feline immunodeficiency virus, and during those studies I got interested in the ways that we measure antibody responses to a range of viruses of companion animals. So for the last 3 or 4 years we've been looking at rabies virus, Rift Valley fever virus and the morbilliviruses.
Okay, and then how did you get to where you are today here working at the CVR? Can you give us a background about your undergrad maybe and your PhD?
Well, I'm actually a biochemist stroke pharmacologist from Strathclyde University. But when I was at Strathclyde University, I decided to work on immunology. I got kind of interested in animal cell culture. So after a PhD in immunology, I then got interested in retroviruses. And I have to confess, I was inspired by a very young Ruth Jarrett who came to give us a seminar on HIV over at Strathclyde University.
And for those who don't know, Ruth Jarrett is also working here at the CVR as a programme leader as well.
You've recently published a few papers about morbilliviruses. So first of all, can you tell us what are morbilliviruses?
Well, most of you will know morbilliviruses from measles, the virus that is in the MMR vaccine. There's also canine distemper virus of dogs. And there's a famous virus, rinderpest, that's now been eradicated. So there's a range of different morbilliviruses. You find them in seals, whales, a whole range of different species.
And what ones in your lab are you particularly interested in?
Well, we were originally interested in canine distemper virus. So there was an ongoing study in the Serengeti looking at the relationship between dogs and the development of canine distemper or distemper in lion populations. Was the virus being spread from the dogs to the lions or from other wildlife species to the lions? And we developed an assay with which we could measure antibodies to canine distemper virus much more sensitively. And once we developed a canine distemper virus assay, we realised we could then extend that to rinderpest, to measles, and to peste petit ruminant, a virus that affects sheep and goats.
So this peste petit ruminant, excuse my French.
It's easy for you to say.
It's called PPRV for short.
Yes.
So it manifests itself in small ruminants. What ways does it manifest itself? What are the symptoms?
Well, the range of I am no veterinarian. I'll say that straight up front. The range of clinical signs. You can detect it with ulceration in the mouth. You'll get diarrhea and wasting disease. It's a significant problem to sheep and goats around the world. Sheep and goats are a very important source of food and livelihood to many people, some of the poorest farmers around the world. And there is a plan to actually eradicate PPRV, following on from the success of the rinderpest eradication programme. So it's important that we identify the reservoirs for PPRV. So if you want to eliminate a virus from sheep and from goats, you have to make sure that there are no hidden reservoirs of the virus lurking somewhere that can start a new epidemic.
And that's something we'll talk about later.
Okay.
It's interesting.
You also mentioned canine distemper virus, or CDV for short, and this is a virus that's widespread in carnivores, including some endangered species. So how do you think this is affecting their populations, and do you think it's a risk?
Well, once we developed an assay for CDV, we were actually contacted by one of the previous people who were on a related podcast, Martin Gilbert, and he works on canine distemper and how it's affecting tigers in Russia, the Russian Far East. And he asked us if we could actually screen a range of samples he'd collected from tigers and from wildlife for antibodies to CDV. So we screened a whole range of samples from the Russian Far East, and we found that yes, we could detect antibodies not only in tigers but also samples from sable and samples from badger, leopard cat. So the virus is actually present in a whole range of different species. There was a paper a couple of years ago in which they proposed changing the name from canine distemper to carnivore distemper, but as we've subsequently found, that would still not cover all the different species. Canine distemper is actually a remarkable virus in that it can infect everything that is exposed to the virus. It's an extremely broad tropism virus.
So you've referenced this assay a couple of times, this new assay that you've come up with. So it's using pseudotypes to study the seroprevalence of morbillic viruses in lots of different animals. As you say, it infects lots of different animals. So first of all, can you tell us what is a pseudotype?
Simplistically, you take the envelope glycoproteins, so that's the proteins from one virus, and you express them on the surface of another virus. So we first started doing this with lentiviruses. It was an HIV-based virus system. So we expressed the feline immunodeficiency virus glycoprotein on HIV, and that allowed us to measure FIV neutralizing antibodies. We then adapted the system to measure rabies neutralization, inspired by studies by Ed Wright and Robin Weiss, and we expressed the rabies glycoprotein this time on a gamma retrovirus, murine leukemia virus. And as we were studying neutralization, we thought, well, why don't we start looking at morbilliviruses? So initially we used morbillivirus glycoproteins expressed on lentiviruses, and the titers were never particularly good. So we adapted the system to look at vesicular stomatitis virus, and the titers that you obtain with vesicular stomatitis virus are excellent. So you're able to measure neutralization against your morbillivirus, but it's using a defective vesicular stomatitis virus.
So just to explain, a virus pseudotype is when you take the glycoproteins from one virus and express them on the surface of another virus. Virus glycoproteins are the proteins that are found on the outside of the virus that recognize and bind to the receptors of the cell surface. It is often these glycoproteins that the host will build an immune response against. Therefore, when you make a pseudotype virus that expresses the glycoproteins of your virus of interest, you can measure neutralizing antibodies against this virus. Brian's group has taken advantage of this technology to make pseudotype viruses that are safe to work with in lower safety laboratories rather than working with the live virus. In this way, they can measure neutralizing antibodies to the different viruses they are interested in.
And what are some of the obstacles that your assay overcomes compared to other assays?
Well, you don't actually have to work with the primary virus. So if you wanted to measure neutralization to rinderpest, you'd have to use live rinderpest. And you can't do that. You can only do that in the UK in Pirbright. So there are very few laboratories worldwide. You don't have to work with a primary biological isolate. So if you take a sample from a tiger in Russia and you want to grow the virus from it, that may also contain a range of other viruses. The animal may have been exposed to rabies, for example. If you're using a pseudotyping system, all you're doing is you're expressing the viral glycoprotein, so it's safe. You can do it in a CL2 laboratory. So it makes the assay system much more accessible to laboratories around the world. And that allows us to measure neutralizing antibodies, for example, against measles without having to grow measles.
And another interesting thing about your assay is that you can take the amino acid sequence directly from a wild type virus and study that on the pseudotype, whereas opposed to live virus systems, maybe they've passaged the virus. What are the problems with passaging the virus?
Well, one of the biggest problems is that if you passage a virus, for example, the Edmondson strain of measles or the Onderstpoort strain of canine distemper virus, those viruses adapt to cell culture and they change the receptors that they use to get into their target cells. And in doing so, that can actually change the neutralization properties. So you might actually not be seeing a biologically relevant neutralizing response when you're performing your assay. There's also a sensitivity issue. If you use a live virus based neutralization assay system, the sensitivity is not that great. So when we use the pseudotyping system, we can actually improve the sensitivity by at least tenfold, and quite often a lot more than that.
And it's also, you've described it as rapid. How quickly can you turn over?
Well, you can read your assay in a couple of days, basically. Some of the live virus neutralization assays can take anything up to a couple of weeks. So it's a couple of days turnover, and you can use, you can stack up plates. You can have piles of 96 well plates, and you can measure a whole range of different samples at the same time. We're only limited by, at the moment, by the plate reader we have. We could increase that to a 384 well plate reader, in which case we could do far more samples. And you can use very, very small volumes of serum. And when you're working on something like cattle, serum's not an issue. But when you're working on something like sable or weasels, then serum does become a bit of an issue. And the smaller the volume, the better.
And another interesting thing you can do with this also is, because you're using plasmids to have the glycoproteins in the future, maybe you could do site-directed mutagenesis to look at specific amino acid changes.
Yes, now that is incredibly easy to do. So as long as the mutations you make do not change how much of the glycoprotein is expressed on the surface of the pseudotype, in theory you can map any epitope you want. All you have to do is make the mutations in your plasmid, then prepare the pseudotypes. So we can actually, for example, with PPRV, we can actually measure the neutralizing response against the 4 different lineages of PPRV rather than just using one lineage. Existing neutralization assays are all based on one lineage, the Nigeria 75 virus. We can actually measure neutralizing responses against primary strains of 4 different lineages.
And with your assay, is cross-reactivity a problem? And if it is, how do you overcome that?
I guess you probably mean cross-neutralization,
Yeah.
or do you mean non-specific neutralization?
If you get neutralization for 2 different Morbelli viruses, how do you know?
Well, we can actually tell the difference between the neutralizing responses. So if you were to submit a sample to a standard veterinary diagnostic laboratory that uses a live virus-based test, they would simply measure neutralization of canine distemper virus, and Onderstpoort strain probably, and nothing else. So you wouldn't actually know whether or not the animal had been exposed to canine distemper virus or another Morbelli virus. Likewise, if there's a non-specific neutralization of the virus, it's difficult to tell whether or not that's caused. Is a low titer really specific? And that was actually one of the reasons why Martin Gilbert contacted us about the tiger samples. They were getting very low neutralising titers. So since we could increase the sensitivity of the test, we could tell whether or not they were real. And then if we compared CDV with PPRV or measles or rinderpest, we could tell if it was specific. So we can actually distinguish different specificities, different species of virus.
And one of the results from one of your papers is that you have higher neutralising antibodies induced by natural infection rather than vaccination. Do you know why that would be?
Well, it might be very simple. It might be that you get higher viral load with natural infection. So there was a study we did in collaboration with Michael Barron at Pirbright, and he had run an experimental vaccine trial, and he was vaccinating cattle with PPRV to see if PPRV would cross-protect against rinderpest. Now, the reason for that is rinderpest has been eradicated, so you want to get rid of all stocks of live attenuated rinderpest. You don't want to have them being held and the possibility of it escaping. So you wanted to see if PPRV vaccines, which are being used widely, would protect against rinderpest. The outcome of the study was that the wild-type PPRV gave far higher titers than the vaccine strains of PPRV, and only with the wild-type could you really detect significant neutralising titers against rinderpest. However, when we used the pseudotype test, we could actually push the sensitivity so far that we could actually detect low levels of neutralising antibody in the animals that were given the vaccine strains of PPRV as well. So they would normally have fallen below the sensitivity of existing live virus neutralising assays.
So PPRV is normally thought to infect sheep and goats. However, in one of your papers you've found seroprevalent cattle, which you describe as an atypical host. Can you tell us a bit more about that?
Yeah, so we used rinderpest, CDV, PPRV as controls for each other. So when we were looking for PPRV, we would use CDV as a control, or we'd use rinderpest as a control, because we weren't expecting to see responses to them. But during the surveys that we started to do, we actually found that there were several of the cattle samples that actually had neutralising antibodies against PPRV, and funnily enough, some of them also had neutralising antibodies against CDV. So that shouldn't be entirely unexpected, because CDV appears to get absolutely everywhere. But it did suggest to us that we will have to be very careful when we vaccinate sheep and goats to eradicate PPRV. Are we going to leave a reservoir of infection in cattle? Because they are susceptible to PPRV. The infection appears to be inapparent in that they don't develop any clinical signs. But the big question is, are there animals that actually will shed virus? And that we don't know at the moment.
Another interesting finding that you found in cattle was that you found 7 that were seroprevalent for rinderpest. So you're not suggesting that rinderpest is alive and well? Or?
Well, what we can say is that the animals absolutely 100% were infected with or exposed to a rinderpest-like virus. The question is, what kind of exposure was that? Was it vaccine? Was there some kind of very old stock of vaccine still being used? Or is there still a rinderpest strain going about that is does not cause any clinical signs? Now, just when rinderpest was actually eradicated, the very last cases that were found in the Meru National Park in Kenya were only found through serological testing. The animals didn't have any signs of disease. It was only through routine screening that they actually picked them up. So it could be that there are still remnants of the original infection going around at a very low level. That we don't know yet. We have extended our studies through a range of other villages now across the Serengeti, and we have found quite a few other samples that are rinderpest positive. Most of those samples come from old animals, which is in a sense reassuring. It kind of suggests it might have been vaccine exposures, but it could also have been exposures to the original strains that were circulating, and they're just the last remaining animals that are still seropositive.
So do you think your lab can aid the World Organization for Animal Health to achieve their goal of PPRV eradication by 2030?
Yes, we think that there's a very important question that has to be answered in regard to whether or not cattle are a major reservoir and can they shed virus back to sheep and goats. And we think we can actually show that. because we've got sufficient sample numbers. We have several thousand samples being collected for other reasons in Tanzania. And if we test both the cattle, sheep and goats for antibodies to PPRV, we have such complex histories of the animals that we can actually work out whether or not the virus is moving from the sheep and goats to the cattle or from the cattle back to the sheep and goats. That's our target at the moment.
And where do you think this research is going to take you next? What's the future plans?
Our future plans, we're actually quite interested in how neutralising antibodies are, what level of neutralising antibodies are being elicited by the MMR vaccine and why we're getting little clusters of measles breaking out. For example, there's a little cluster of measles in Lothian at the moment, maybe 18 cases, 16 or 18 cases. And is that simply because titers are low, or are there some genotypes that are able to resist neutralisation sufficiently to get over the threshold to establish a new cluster of infection? And we have some evidence to suggest that that is actually the case, because there are some genotypes, even though they've only got a few amino acids different, they do resist neutralisation better than the vaccine strain. And so it is possible that some of the little clusters that we're detecting are a combination of two things. Not sufficient coverage of the vaccination amongst the population, dropping titers because people were vaccinated a long time ago, but also possibly because the genotypes that are coming around now are slightly different and they're not all being neutralised by the titers that we raise with the MMR.
So with your work on morbilliviruses, what would be your take-home message for the listeners?
What is my take-home message? I think we should revisit many of the old assays that we used to use and the old questions that we thought were resolved 20, 30 years ago. Because technology has moved on so far now, we shouldn't just think of everything as being an opportunity to sequence something, get some nucleic acid and sequence it. There is actually an important biological question sometimes that you can answer using an updated technique. So the technique that we've put a lot of effort into, the pseudotyping technique, actually allows us to go back and revisit neutralisation in a much more powerful way. We can look at cross-neutralisation, we can look with much more sensitivity. So you'll come across many studies which said that we couldn't detect neutralising antibodies, therefore it must be a cellular response that mediates immunity. Well, that's simply not correct, because if you look a wee bit harder, you can actually find a neutralising response. So we should reevaluate many of the conclusions that we made, decades ago.
And through all of this work, you've formed a lot of collaborations. So we've heard about work that you've done with Pirbright down in England, and also work that you've done with other institutes here at the University of Glasgow, Sarah Cleland. Do you have any advice for other people who want to set up collaborations on how to do that?
Don't be shy. Just phone people up or email them, because most people will collaborate with you. One of the most fun collaborations we've had in the last year was with Ed Ramsey, who was vaccinating tigers. We were trying to figure out whether or not existing canine vaccines would protect tigers from field strains in Russia of CDV. And we came across Ed Ramsey, who was in the University of Tennessee, and he had been actually vaccinating tigers in the US, because they have loads of tigers in the US and they have to rehome them. So they have a tiger rehoming facility. And to make sure that they don't get distemper, they vaccinate them with existing dog vaccines. So we were able to analyse samples from tigers to see if the sera that were raised in those animals would cross-neutralise Russian field strains of canine distemper. And the only reason we could do that was because we had access to the glycoprotein genes from the viruses in Russia, and we had the sera from the tigers in the US. We could put the two together and we could get an answer. And the answer is yes.
So a question that we really like to ask is, if you weren't a scientist, what would you be?
If I wasn't a scientist, I'd probably be running an RSPB centre somewhere, or I'd be a gardener. I'd be Monty Don.
So that's another episode of Contagious Thinking over. Today we've learned about morbilliviruses and how they may be more widespread than we realised, infecting many different species of animals. Brian has also explained in detail about the pseudotyping assay that was developed to detect neutralising antibodies to these viruses and all the positive aspects to it, from being able to work with these dangerous viruses in a lower safety level laboratory, making them more accessible to be studied, to how the sensitivity of the assay has been increased. The finding of animals that have been exposed to a rinderpest-like virus brings up questions of whether there is an old stock of rinderpest vaccine being used, or is there still a rinderpest strain going about that doesn't cause clinical signs? More investigation needs to be carried out to answer these questions. The World Organization for Animal Health wants to eradicate PPRV by 2030. The results that Brian's group has found show that if a vaccination strategy is implemented, it will be important to also consider other animals such as cattle, which may act as a reservoir for the disease, even though they don't show any clinical signs of infection. Thanks very much to Brian for agreeing to speak to us about his recently published research, and thank you very much for listening. I'm Joanna, and you've been listening to the CVR's Contagious Thinking podcast. See you next time.